and invite Emanuele Pio Barracchia for his presentation.
Okay, thank you.
He is from the Universidad de Bari Automóvil too, and he's joint work with the CINI and CNR and the Joseph Stefan Institute too. Please share your, okay. Go ahead, please start your presentation. But your microphone is not working.
Okay, can you hear me now?
Yeah, now it's good.
Okay. Good evening, everyone. My name is Emanuele Pio Barracchia, and I am a PhD student in computer science at the University of Bari. And today I'm going to talk about LP-HCLUS, a novel tool for the prediction of relationships between non-coding RNAs and human diseases. So many non-coding RNAs play a key role in human diseases. For this reason, in literature, there are many works that try to study relationship between non-coding RNAs and diseases in order to understand the molecular mechanism underlying these diseases. To reach this goal, many works exploit a network structure to represent biological entities such as genes, non-coding RNAs, diseases, proteins, and so on, and their relationships. However, there are some limitations. First of all, many of them works on homogeneous networks that are networks with only one type of biological entity. Other works are limited to a set of biological entities, and many of them does not take into account a feature associated with the nodes. So the goal of this work is to develop a new system that allows us to predict previously unknown association between non-coding RNAs and diseases that is able to analyze heterogeneous attributed networks that are networks where there are different biological entities and can be with their features, and this network can be of arbitrary structure. And we want also to provide biologists with a tool to help them to make to conduct a more focused research at different level of granularity. So we propose LP-HCLUS that can analyze the network with different level of detail, can exploit information combined by the feature associated with the nodes that are the biological entities in the network. And it is able also to catch the multiple roles that a biological entity could play in the network. So the basic the ideas underlying LP-HCLUS are two. The first one is that diseases influenced by the same non-coding RNA can share group of non-coding RNAs, and on the other hand, non-coding RNAs that cause the same disease may collaborate to develop a new disease. So LP-HCLUS, for this reason, exploit a multi-type hierarchical clustering that groups together diseases and non-coding RNAs that are related in some way. This is the workflow of the system. In particular, the first step is the estimation of the strength of the relationships. Given an heterogeneous network, LP-HCLUS will first identify the target types that are those node types that are relevant for the link prediction task, in this case, non-coding RNAs and diseases, and the task relevant types that are the node types that are considered only during the identification of the relationships between target types. From the heterogeneous network, LP-HCLUS will identify metapaths. A metapath is a sequence of nodes that follow the same sequence of edge type. Given an non-coding RNA N and a disease D, the score associated with the pair is computed as the maximum score observed over the metapaths. In particular, given a metapath P, if there exists a sequence in P that connects N and D, then the score will be equal to 1. Otherwise, it will be computed as the maximum similarity between the sequences that start with N and those which end with D. The second step is the construction of the hierarchy of cluster. During this step, LP-HCLUS will identify clusters that are heterogeneous and overlapping because they will contain different biological entities that and it will catch the multiple roles that they can play in the network, and also and it will construct a hierarchy of these clusters, and it will make easier to understand by researchers. These clusters are then exploited during the prediction step. During this step, for each level of the hierarchy, LP-HCLUS will look for new association in the clusters, and in the case of the association can be derived only from one cluster, the final score associated with it will be equal to the coherence score of the cluster. Otherwise, if the association can be derived from different clusters at the same time because the clusters are overlapping, the coherence score are combined following one of the implemented combination strategies that are maximum, minimum, average, and evidence combination. In particular, evidence combination has a main idea that if the association can be derived from multiple clusters, it has more probability to exist. So this is an example of the prediction step. And as we can see, we can spot two different new relationships. The blue one that can be derived only from C1. So in this case, the final score will be equal to the coherence score of C1, while the red one can be derived from both C1 and C2. So in this case, LP-HCLUS will adopt one of the combination strategies. We conducted several experiments comparing LP-HCLUS with HCLUS-2, that is a competitor that, similar to LP-HCLUS, construct a hierarchy of possible overlapping clusters, but it is limited only to 2 different types of nodes. ncPred, that is a state-of-the-art method specifically designed to predict non-coding RNA disease association, but it does not take into account the features associated with the biological entities. And LP-HCLUS NLP, that is a modified version of LP-HCLUS without the clustering and the prediction step. So the score associated with each association are the one computing on the basis of the metapaths. We used 2 different datasets. The first one is HMDD, that is a dataset containing information about diseases and microRNAs and their relationships. And as we can see, LP-HCLUS, in particular, exploiting maximum as a combination strategy, is able to outperform its competitors. And we also construct a dataset combining state-of-the-art dataset containing information about diseases, non-coding RNA, target genes, and their relationships. Also, in this case, LP-HCLUS achieved the best performances. We also conducted a qualitative evaluation of the results. As concerned HMDD, we compared the association predicted by LP-HCLUS against those one experimental validated and introduced in the updated version of the dataset. As we can see in the image, they are depicted only the top 100 association predicted by LP-HCLUS, and we highlighted with the dark green lines those associations that have been introduced in the update and the recent version of HMDD. As concerning the integrated dataset, instead, we compared the output of LP-HCLUS with the data in the MDR database. In particular, we focused on the H19 disease association, and we found a perfect 33 perfect matches with also similar scores. This is only an example. And H19 is a long non-coding RNA that participates in the epigenetic regulation of many processes of diseases. LP-HCLUS found an association between H19 and bone diseases, but this association is not present in the literature and is not even reported in the MDR database. Bone diseases can have different origins and can have origin also from the hyperfunction and the hypofunction of endocrine glands. But the output of LP-HCLUS and data in MNDR confirm the existence of association between H19 and diseases of the endocrine glands, as you can see from these images. This indicates that H19 can have a relationship with the endocrine glands function and therefore can have a relationship with bone disease. This finding is only an example of how the ability of LP-HCLUS to catch the network autocorrelation phenomena and to exploit information combined by the network structure can help biologists to formulate a new hypothesis. So, in conclusion, we propose a LP-HCLUS that is able to work with heterogeneous attributed biological networks. From a quantitative point of view, LP-HCLUS is able to achieve good performance. And from a qualitative point of view, we found that several associations predicted by LP-HCLUS have been subsequently experimentally validated. And the example reported on H19 and its potential role in bone disease demonstrate that LP-HCLUS can be a powerful tool for researchers to better customize their experimental investigations. For as future work, we want to expand the system in order to predict also the direction of the link, but we have to deal with the cause-effect phenomena. And we want also to evaluate the performance of LP-HCLUS in other domains, for example, tissue-specific dataset or pathological specific conditions. All the data, the system, and the results are can be accessed via the link in this slide. Thank you for your attention.
Thank you, Emanuele. Your presentation was very, very interesting, and we have time for a few questions. If someone likes to do directly using the microphone. I actually have a question. Normally with this kind of approaches, I have the idea that how correlated is the real biology with the computational biology result. How is that result compare, if you compare with another techniques for to do that? You have some idea? Because you showed only some points of the approach, give some ideas of the possible relations, but how you can check in your models if the biological part is correlated and you have a good accuracy and good precision.
We need an help of a biologist because we can develop a method that can help a biologist, but I think that the final decision have to be made by a biologist, because we cannot substitute a biologist and say, no, our method find this and this is true.
Yeah, you're right. I mean, if I am a biologist and I receive your information, I hope that information has a big probability to be a target, really. Because I have to put some experiments and try to check what you are giving me through the computational method. My point is how you can give me some kind of precision or accuracy of your method, but allow me to take that decision in base of that statistical or whatever other measures you can use in the method. And how you compare your method with other methods, because in this process, normally you find many different approaches. And almost any of them are, has a benchmark or something like that.
Okay, for now, the experiments have been conducted using part of the data we have as a testing set. So for now, it is the only way to test in terms of accuracy our time of recall, because accuracy is difficult to compute because we need negative samples, and in this domain is difficult to have negative samples. And but what we can do and what LP-HCLUS does is give to biologists a list of predicted association from the most probable to the least.
I don't see any other questions. Thank you very much.
Hi there, can you hear me? I'm Pratim.
Biologists, yeah.
Go ahead, please.
Yeah, can I ask the question?
Yeah, please.
Yes, please.
Okay, hi. It was a great presentation. I mean, I joined late. I'm sorry. I just have two specific questions. You know, let's say I give you a set of, you know, even in the H class, if I give a set of pathways, which are kind of in a specific format in which H class accepts it, you know, can it kind of, if I can perturb, let's say, you know, why the point from where I'm speaking is because there are many pathways which in bacterial population which becomes drug resistant in subsequent you know, time. Can I kind of perturb a particular pathway and try to figure out, based on the weightage that you put on the pathway, that what are the alternative routes that are being opened under different perspectives, sort of stuff?
So the question is, if you give an input to some pathways, if you can get prediction from them, right?
Yeah, let's say you said that, you know, it gives the biologist a list of pathways with a from the most probable to the least probable thing, right?
Okay, at the moment, LP-HCLUS accept an input a database containing all the information about the biological entities. But maybe it could be an extension of the system.
Okay.
To test.
I'm sorry, you were breaking up.
Can you?
I think I could not hear you. Okay, I'll get back to you later. Thank you very much for answering.
Thank you. Thank you for your question.
Bye. Take care.
Emanuele, thank you very much for your.
Thank you.